Hello, biology superstars! Welcome to the SL content for A1.2 about nucleic acids. We have 10 points of information to get through, so let's get started. The main ideas for today are DNA and RNA are responsible for carrying genetic information. So these are the nucleic acids that we're primarily going to be studying. The basic repeating structure of DNA and RNA is called a nucleotide. We'll talk about the structure of a nucleotide and its three component parts, which are a nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. This is something that you need to know how to draw a generalized diagram of, so we'll go over how to do that. Nucleotides look slightly different in DNA and RNA. You are responsible for knowing about the differences, and we'll talk about them. And similarity of DNA and the genetic code used between different living organisms is used as proof for the theory of evolution. And this is where we get into that idea of unity and diversity that is really central to the whole theme that we're studying. I'm going to see if I can get my pen going. It seems like maybe I cannot. I think it'll work better on the next slide. All right, but so this is theme A, unity and diversity. All right. First point of information. DNA as the genetic material of all living organisms. Some viruses use RNA as their genetic material, but viruses are not considered to be living. I would just make a little note of this fact. This could come up as a multiple choice question, for example. And if you're in the HL course, you will talk about RNA as probably the genetic material of the very first living organisms, but in the origin of cells, which is HL only content. But at this point, we really say that DNA is the genetic material of living organisms on the planet. And we'll talk a little bit about the basic structure of DNA. So DNA is a type of nucleic acid. I'm going to see if my pen, come on, pen. Oh, yay. All right. So DNA, we'll change to orange. Why not? A type of nucleic acid. You're looking at a variety of different views of a DNA molecule here. This is twisted up into a double helix. This is that same double helix polymer, but untwisted. And then this is like a close-up view that shows you all of the different nucleotides that are used in the composition of a big, long polymer of DNA. So these are all monomers. Let's see. DNA is important because it can be passed from parent to child, so it is used for heredity. And nucleic acids are one of the major groups of biomolecules. So along with carbohydrates, lipids, and proteins, nucleic acids are like molecules that are really present in all living things. All right. Next point of information is components of a nucleotide. So this basic monomer structure, the building block of a DNA polymer, you need to know how to draw this. You can use circles and pentagons and rectangles to represent the relative position of phosphates and pentose sugars and bases. And we will go over how to do that on the next slide. So when you, let me actually change my pen again. Not yellow, blue. I like blue for this. Okay, so nucleotides are the building blocks of nucleic acids. DNA and RNA are polymers, and nucleotides are their building block monomers. Those nucleotides consist of three parts: a ribose sugar, This is our pentagon. A phosphate group. This is our circle. And a nitrogenous base. This is our rectangle. So if you are asked to sketch a DNA nucleotide or an RNA nucleotide, labeling all relevant components, you can start with any part of it you want. I think I'm going to start at the sugar. So a pentagon is a 5-sided structure. You have to draw the phosphate group coming off the edge of the pentagon in a certain way. So if you were to think of this, all of these numbers as like the corners of this shape, we'll go over this more in another slide. You want to draw the phosphate group coming off the, like a little straight edge that makes a 5th corner above the left-hand side of the pentagon. At the first corner, that's where you want to draw the rectangle. And remember, you need to also be able to label these. So the sugar is the pentagon. This is our number 1. The phosphate group is the circle. This is our number 2. And the nitrogenous base is the rectangle. This is our number 3. If you were doing this on an exam, you'd want to write the actual words. I'm just like trying to save space by annotating with the numbers. And let's talk about how these nucleotides are held together in a polymer of DNA or RNA. And to talk about that, we are going to bring up sugar-phosphate bonding. So sugar-phosphate bonding makes a continuous chain of covalently bonded atoms in each strand of DNA or RNA nucleotides, which forms a strong backbone in the molecule. Remember, we already talked about covalent bonds in A1.1 about water, and they come back up right here. The covalent bond that is present in a DNA molecule is so important that it actually has a special name. You call maybe I'll change my pen again. That's kind of fun. You call, well, maybe. Come here. No, I want that blue. Okay, so you call the covalent bond that is present between the phosphate group and the sugar, so I'm looking right there. You call it a phosphodiester bond. And it seems like a big confusing word, but I actually sort of think it's one of those words that is helpful to know, because once you understand what it means, it can be helpful in like remembering what it does and where it is. So phospho means that it is close to the phosphate group. Here is the phosphate group. Di means that there are 2 of them. So you'll notice like the phosphate group is bonded to this nucleotide. There's also a phosphate group bonded to the bottom side of the nucleotide that technically belongs to this next nucleotide down. So in the sugar phosphate backbone, there's always a bond forming like between the phosphate group of one nucleotide and the sugar of the next nucleotide. This phosphate group, if the chain kept going, would be connected to another sugar on the other side, and it keeps going and going. And then an ester bond, for our purposes, I'm going to say that this is when an OH, a hydroxyl group, bonds with an H. Your IB chemistry teacher might get a little bit mad at me, but for what is happening here, that is sufficient explanation. If you're not in IB chemistry, don't worry about it. But you can sort of see, like here, there's an O at this bond. You can maybe imagine there's like some hydrogen atoms that are also involved in the bond. So that is the sugar phosphate backbone and the covalent bonds that make it so strong. When you're looking at the whole molecule, you can also notice that there are hydrogen bonds forming between the bases in the middle that make like, you call them rungs, because if you look at this and it looks like a ladder, this is like the rung that you would climb on. So the next thing we'll talk about is actually the bases and how they're named and how they're held together, and a little bit more about the middle of the DNA ladder. So 1.2.4 is the bases in each nucleic acid that form the basis of a code, and the IB says that you should know the names of the nitrogenous bases. I would be surprised if IB asked you to like state all the names of the bases. They could. I think this might be more likely to come up as like to recognize the names in a multiple choice question. But theoretically, they could ask you to write out any of these names: cytosine, guanine, adenine, or thymine. And if you're talking about RNA, uracil instead of thymine. So the next slide just lines these bases up. The bases that are in DNA versus the bases that are in RNA. Here they are. And we'll talk a little bit more about the different bases when we talk about our differences in DNA and RNA over the next few slides. So I think let's just leave it there for now and keep going. Next thing, RNA as a polymer held together by nucleotide monomers. And students should be able to draw and recognize diagrams of the structure of single nucleotides and RNA polymers. This is a little bit where that uracil thing comes in. So like if you have a picture of a nucleotide where the base that is used is uracil, so like let's go here, and they tell you that the rectangle nitrogenous base is uracil, you are expected to recognize that this has to be a nucleotide of RNA, because only RNA has this nitrogenous base. If you are asked about RNA as a polymer, I would say you should be focused on the fact that RNA as a polymer only has one strand. Like you call DNA a double helix because there's two of these sugar phosphate backbones, one on each side. RNA only has one sugar phosphate backbone. So it's a single stranded molecule. And we'll get to that again when we talk about differences between DNA and RNA. But in terms of drawing and recognizing a diagram, those are the things that I would keep in mind. Uracil and single stranded. U and one strand. All right. So if we're going to talk about drawing and recognizing RNA polymers, we also have to talk about drawing and recognizing DNA polymers. And DNA is a little bit more complicated, and they go into a little bit more detail. Let's go purple, I think, for DNA. If I can get to the purple. There we go. So DNA as a double helix made of two anti-parallel strands of nucleotides with two strands linked by hydrogen bonding between complementary base pairs. This point of information sort of brings together a lot of things we've talked about already: the hydrogen bonding, the complementary base pairs, the double helix. The anti-parallel thing we'll get to in the notes, and nucleotides we've already discussed. But this assessment statement gives you some more specific information about exactly what you need to be able to do with this information. So in diagrams of DNA, students should draw. This is something you need to be able to draw. The two strands anti-parallel, again, we'll go over how to do that, but are not required to draw the helical shape. So you can draw it flat, not all twisted up like this. Students should show adenine paired with thymine and guanine paired with cytosine. Students are not required to memorize the relative lengths of purine and pyrimidine bases or the number of hydrogen bonds, except you are a little bit for HL. We'll get into that in the HL video if you're in the HL course. But let's look at the basics of this double helix and explain this anti-parallel thing. So a non-helical DNA drawing could look a lot like this. Yes. The double helix means you're showing that there are two sides. The anti-parallel thing, obviously anti-parallel is the opposite of parallel, and the anti-parallel thing has to do with the direction that the sugar phosphate sides are facing. So if you look at these molecules, the little phosphate groups are on different sides, depending on whether you're looking at the left hand or the right hand part of the picture. So let's start with the left-hand part. It's almost like this side of the molecule is running up. There's a phosphate group at the top, right? Like this is what makes me think this one's going up. If the strands were parallel, the other one would be doing the same thing, right? That's what parallel means. But the other one is going the exact opposite direction, which is why we call DNA strands anti-parallel. This one is going down. The little phosphate groups are pointing down. And so are the points of the sugar. I think this is like another thing that makes it relatively easy to remember what anti-parallel means, is you can follow the sugar points and think about if they're pointing up or down. So remember we said before that the idea that the carbons in the sugar can be numbered is important. This comes back a little bit here. This is like probably a little bit more than you need to know for SL. But when you draw one of these molecules, you have the oxygen at the point, and then the 5 carbons, 1, 2, 3, 4, 5, around the side. The loose end that has a phosphate group on it is the 5' end of the molecule, because the phosphate group hangs off the 5th carbon. The loose end that does not have a phosphate group on it is called the 3' end, because this is the 1, 2, 3rd carbon, and that's the loose end here. So when you're drawing this DNA double helix, you want to make sure that you keep the sugars pointed the right way. So like that sugar is pointing up. 1, 2, 3, 4, off the 5th. Oh no, I don't know why it does that sometimes. I'll just erase this like weird rogue line and put the phosphate group here. And then on the other side, you want to make sure that the sugar is pointed down. And you still have to have the base in the middle, because the bases need to meet up. You counting backwards here. This is 1, 2, 3, 4, 5, and the phosphate group goes here. So that is like a not very good, but hopefully helpful drawing of what anti-parallel strands of DNA would look like. All right. The next thing that we need to talk a little bit more about are other differences between DNA and RNA. So if you're asked to contrast these two molecules, you need to, let's go pink. You need to talk about the number of strands present in the polymers, the types of nitrogenous bases that you might encounter, and the type of pentose sugar. Something that you should be able to sketch is the difference between ribose and deoxyribose sugar. We'll talk about that on the next slide. And students should be familiar with examples of nucleic acids. Those examples are basically ATP, DNA, and RNA. All right. Let's go to the next slide. We'll do some comparison between DNA and RNA, and specifically this ribose and deoxyribose sugar thing. So type of sugar, number of strands, and bases present are all just easy to sort of study in table form. These are all things we've talked about already in this lecture, except the ribose and deoxyribose sugar, which we're going to go over now. The position in cell I'm leaving in here because when you start thinking about the use of these molecules in replication and gene expression, which will be the next point of information, it's helpful to think about where they are in the cell and whether or not they are able to move around in the cell. All right, but first, let's talk about the ribose and deoxyribose sugar thing. All right, I'm going to label, hopefully a little bit more clearly here, these 5 carbons that are in this ribose sugar, or in this pentose sugar, this 5-sided sugar. If the sugar is ribose, like in RNA, that means that it is fully oxygenated. Like there is a bunch of oxygen in all of these different carbon positions. If the sugar is deoxyribose, that's exactly what it sounds like. It means that some of the oxygen has been taken out of the molecule. Specifically, the oxygen at the second carbon position has been removed, deoxygenating the sugar molecule. Deoxy, O at carbon 2 removed. Okay. So if you were asked to sketch just a regular ribose sugar, you would sketch the one that is here. If you were asked to sketch a deoxyribose sugar, you could sketch the same one, except you could just take out this whole hydroxyl group, because the IB takes it for granted that you understand that, like, any place where there's not a hydroxyl group, there's a lone hydrogen atom. Don't worry about it too much. This would be the picture if it was deoxy. Deoxy because of this, or regular fully oxygenated ribose would look like this. All right, let's get back to this idea of position of these molecules in the cell. And this oxy-deoxy thing is actually important in this. The deoxygenation of the ribose sugar in DNA lends a lot of stability to the molecule. It's part of the reason it can form double-stranded, and part of the reason that it gets stuck in the nucleus, because it is bigger. RNA is more reactive, more likely to break down, and only forms that single strand because it has this oxygen in it. So these single-stranded molecules of nucleic acids are able to travel throughout the cell from the nucleus to the ribosome, which makes them very useful in gene expression. And the purpose of these molecules is what we will get to in the next point of information. So A1.2.8 deals with the role of complementary base pairing in allowing genetic information to be replicated and expressed. And students should understand that the complementarity is based on hydrogen bonding. And this is a new word, to me at least, in the IB syllabus. It was in there before, I didn't notice it, and I think it's so funny. But it's like a legitimate word to describe the complementary nature of the base pairs, but I don't know, it just sounds very unexpected to me. So, but sure, let's talk about it. The hydrogen bonding is super important, and how the complementary base pairing allows genetic information to be both replicated and expressed. All right, so we talked a little bit about the complementary bonding, the complementary base pairs already. You can see it present in this picture. A and T and C and G are together when the molecule is DNA. Here, you can see mRNA matching up with part of the DNA strand, and here you'll notice that A pairs with U. But this idea that the pairing can occur is important for the 2 processes that DNA and RNA are most involved in: replication and gene expression. So let's talk about replication first. The process of DNA replication is semi-conservative, which means that when DNA is going to replicate, It unzips at the center. All those weak hydrogen bonds get broken, and new strands of DNA can be constructed based on the information contained in just half of a strand. So if you have this part of the DNA molecule that I just boxed off, you know exactly what the other side looks like, and you can make it. You can replicate the DNA. I'm going to just like erase this to make everything a little neater. But that idea, I'll even rewrite that, I think. The semi-conservative nature of replication, semi-conservative. The reason that that can happen is because of complementary base pairing. Gene expression also relies on complementary base pairing, but relies on complementary base pairing more between DNA and RNA. So if there is a bunch of information in DNA, an RNA molecule can form a complementary strand, which is actually called a complement, and take that information to a ribosome to make a protein. Both of these concepts are like very important in biology and come up a lot in other sections of the textbook. Replication is D1.1 and protein synthesis is D1.2. So consider this like a very brief introduction to these ideas, which will be revisited at other times in the syllabus. But here we're really just looking at like the structure and function of a nucleotide and how that structure and function allow for these two very important processes to happen. All right. The next point of information has to do with the diversity of possible DNA base sequences and limitless capacity of DNA for storing information. There is a typo in here. Go yellow. I'm feeling yellow. We'll see if you can see it. Yeah. All right. There shouldn't be two ofs. Diversity by any length of DNA molecule and any base sequence is possible. Enormous capacity of DNA for storing data with great economy. So that means like you can have any length of DNA molecule. Humans, the length is 6.4 billion base pairs. Some sort of small genome bacteria, there's only 50,000 base pairs. In a lot of species of flowers, there are like even more base pairs than there are in humans. Tulips have between 32 and 35 billion base pairs. So there's no set length that a DNA molecule has to be. And within that DNA molecule, you can have any base sequence, like the one side of the strand could go A, T, C, G, or it could go A, G, G, C, or it could go G, 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 G. So that gives DNA a lot of sort of flexibility to store data in a very compact way just by switching the order of the base pairs. So since there's 4 base pairs, they say that in, like, for example, in humans, there is 4 to the 6.4 billion possible sequences of DNA that could be present in the organism because the bases can go in any order. And it turns out, I mean, when you look at a genome, that's not like strictly completely true. There are sections of DNA that are like pretty much repeat between all human beings, but just for the process of imaginatively thinking about it, and also for the process of like this particular mathematical equation sometimes comes up on the IB exams. DNA has 4 to the n exponent, and n is like whatever number of base pairs the organism has. That's how many possible sequences of DNA there would be. All right. And the last point of information for the SL content is conservation of genetic code across all life forms as evidence of universal common ancestry. Students are not required to memorize any specific examples. I love when I see this in the assessment statements, because it means that you should just appreciate this as like a broad concept. Across all species, codons code for the same amino acids. And again, we'll talk more about this in protein synthesis, D1.2. It's like a whole subsection of the textbook. Evidence that primitive genetic code was formed in LUCA, which stands for the Last Universal Common Ancestor. I'm going to see if I can get my pen going. Last Universal Common Ancestor. And preserved throughout different life forms. So the same amino acids that are present in LUCA are present in all bacteria, all archaea, and all eukaryota that are found in modern day life. All right, and that brings us to the end of the notes for the SL content on A1.2. I hope this was helpful, and I'll see you in the next video.